Hello and welcome to this program. Today we'll talk about important features and life history of Sacchromyces, and the objectives of this study are to know about the discovery, occurrence and importance of the Sacchromyces, to study the morphology and ultrastructure of the yeast cell, to study the physiological characteristics of this yeast and to present a preliminary exercise for their laboratory culture, to explain at length the types of reproduction in Sacchromyces to comprehend the overall life cycle of this fungus. Sacchromyces belongs to the heterogeneous group of yeasts. They were first described by Anton van Leeuwenhoek in 1680 and were characterized by Louis Pasteur in 19th century as the agents responsible for alcoholic fermentation. Yeasts ferment carbohydrate, hence the name Sacchromyces. The yeast cells were the first to reveal enzyme activity in vivo, and the term enzyme has a Greek origin meaning in yeast. The term yeast has no taxonomic significance as there are Phycomycetes yeasts, Ascomycetes yeasts, Basidiomycetes yeasts and also the Deuteromycetes yeasts. The systematic position of this genus after Alexopoulos and Mynes is given as Kingdom Mycete, Division Amstegomycota, Subdivision Ascomycotina, Class Ascomycetes, Subclass Hemiascomycetidae, Order Endomycetales, Family Sacchromycetaceae, Genus Sacchromyces, Species Cerevisiae, Carlsbergensis, Bailey, Pasteuriana, Diastaticus, Pyriformis, Fermenti, etc. Sacchromyces is one among several genera of Ascomycetes yeasts. It belongs to Protoascomycetes, which consists morphologically simple unicellular and non-mycelial Ascomycetes and which lack the dikaryotic stage, the ascogenous hyphae and as well as ascocorps in their life cycle. But at the same time, Sacchromyces are considered recent in their origin from multicellular and mycelial ancestors via retrogressive evolution. The formation of pseudomycelium during fast budding and that of a true mycelium in presence of penicillin is taken as an evidence of their retrogressive evolution. A thorough taxonomic revision of Sacchromyces has led to their reclassification. Van der Waalt, 1974, noted 40 species of Sacchromyces, but according to Lauder, many of them have been reclassified. For example, Sacchromyces varum is now Sacchromyces cerevisiae variety varum, and Sacchromyces ellipsoides is now Sacchromyces cerevisiae variety ellipsoides. Similarly, Sacchromyces fragilis is now Cluveromyces marcianus, while Sacchromyces roxae and Sacchromyces mellis are now Zygosacchromyces roxae. In a nutshell, some species have been ranked to separate genera and some different species have been clubbed into a single species. As a result, only 30 species of this genus are well recognized, and out of these, the Sacchromyces cerevisiae is the best known. It is the best understood eukaryotic cell and have been the model organism in the study of cell biology, genetics and cancer. Occurrence. Species of Sacchromyces are well distributed. They are particularly abundant in substrates rich in sugary substances containing small amounts of nitrogen and sulfur, such as nectar of flowers, the surface of fruits like grapes, date palm, and exuded sap of plants. They also grow in animal excreta, in milk, on the vegetative parts of plant, in association with insects, in soil and a number of other habitats. One species, Sacchromyces cerevisiae variety ellipsoides, occurs on grapes in nature. When the fruit drops in autumn, the yeasts pass into the soil where they overwinter. In the next spring, they are splashed back by the rain on the plants. Importance. Most yeasts used industrially belong to the genus Sacchromyces, especially the diploid and polyploid races. Bakers and brewers employ Sacchromyces cerevisiae in their industries. The use of Sacchromyces as a leavening agent for bread originated in Egypt about 6,000 years ago and spread slowly from there to the rest of the Western world. The fermentative properties of yeast are used in the baking and brewing industries. In the former case, the desired product is carbon dioxide, which is used to lighten the dough, and in the latter, it is alcohol. The universal employment of this yeast in baking and brewing has further resulted in the establishment of another industry, the commercial preparation of yeast cake. Yeast cake is prepared by mixing and compressing a large number of yeast cells with certain inert matter like starch and cut into cubes, which after drying are sold as yeast cakes of commerce, so often used in alcohol industry and in the home. Because of their high vitamin B, complex content and digestible compounds, proteins, fats, carbohydrates, enzymes, etc., yeasts are particularly valuable as food, as also in the treatment of certain skin disease. When purified and dried at 125 degrees Celsius, they are marketed as yeast white tablets or converted into products like Marmite. Iron added to this yeast is marketed as ironized yeast and recommended for the people with iron-poor blood. Sacchromyces cerevisiae is also genetically engineered to produce a variety of proteins, including the hepatitis B vaccine. It is also used for the extraction of enzymes, invertase and zymase. Extract is used for the preparation of culture media in microbiological laboratories. Many species of Sacchromyces used in the production of different alcoholic beverages are Sacchromyces cerevisiae variety Uvarum, substrate barley malt, product beer, Sacchromyces cerevisiae, Sacchromyces carlsbergensis, substrate molasses, product rum, Sacchromyces cerevisiae variety ellipsoides, substrate grapes, product wine, Sacchromyces cerevisiae, substrate grain mash, product whiskey. Several species of Sacchromyces are also useful in dairy industry, but in the manufacture of grape juice, sweet cider and soft cheeses, the yeasts are harmful and hence must be excluded. Such and other yeasts that are harmful in any way and those that find no use are called as wild yeasts. Shape and size. Sacchromyces include small, non-mycelial, unicellular and uninucleate fungi. They vary in shape between the species and even within the species. They are polymorphic, that is, showing different shapes even in the same culture depending upon the nutrition available. They may be globose, ovoid, elliptical, apiculate and rarely elongated or rectangular. Cells of Sacchromyces cerevisiae measure 6 to 8 into 5 to 6 microns. This yeast has separate haploid and diploid cell populations, and the haploid cells are relatively smaller than diploid cells. The smaller haploid cells, also called dwarf strain cells, are spherical in outline, while the larger diploid cells, also called the large strain cells, are ellipsoidal. The petite mutants of this yeast are still smaller in size. Individual yeast cells appear hyaline, but when grown on solid media, they produce colonies that may be white, cream-colored, pink, or tinged with brownish pigments. Colony characteristics are useful in the taxonomy of yeasts. Physiological characteristics are also utilized in determining yeast species. Laboratory cultivation of yeast is very easy and much like that of bacteria. The yeast can be grown in the laboratory by adding a bit of yeast cake to an aqueous solution of molasses or pasture solution in a flask. The flask is then plugged with a cotton swab and placed in a warm place at a suitable temperature. Within a day, millions of yeast cells will develop in the solution. They also grow on solid media. Sacchromyces cerevisiae is extensively cultivated in bakeries, breweries and distilleries for commercial purpose. Pasteur's solution and its composition. Water is equal to 419 grams, sucrose 75 grams, ammonium tartrate 5 grams, potassium sulfate 1 grams, calcium sulfate 0.1 grams, magnesium sulfate 0.1 grams. It is difficult to differentiate yeast colonies from bacterial ones on agar plates, and the only certain way is by means of microscopic examination of the organisms. Most young yeast colonies are moist and slimy, but many appear mealy. Physiological characteristics. Sacchromyces comprise mostly saprophytic species and some species are also osmophilic. Yeasts are oxidative, fermentative or both. Most of them are obligate aerobes, with a minority of these as facultative anaerobes and only few like petite mutants are obligate anaerobes. The diploid cells are physiologically more active than haploids. The oxidative yeasts may grow as a film, pellicle or scum on the surface of a liquid medium and they are called film yeasts. Fermentative yeasts usually grow throughout the liquid medium and produce carbon dioxide, and the strains of these yeasts in unshaken cultures fall into two principal subtypes. Top yeasts, which are fast-growing, vigorous fermenters acting best at high temperatures around 17 to 21 degrees Celsius and rapidly produce carbon dioxide. The scents clump and are brought by this gas to the surface during fermentation. They are used for making heavy beers with high alcohol content such as English ales. For example, baker's yeast and the yeasts used in ethanol and wine production. Bottom yeasts, which are slow-growing, slow fermenters acting best at lower temperature between 10 to 15 degrees Celsius. They produce carbon dioxide at a slower rate and hence the cells settle to the bottom of the fermentation vat. They produce lighter beers of low alcohol content of the type commonly used in USA. For example, Sacchromyces cerevisiae variety Verum and Sacchromyces carlsbergensis. Yeasts live in optimum temperature of 25 degrees Celsius to 30 degrees Celsius to the maximum of about 35 degrees to 47 Celsius. They thrive best in acidic media at the pH of 4 to 5 and do not grow well in alkaline medium unless adapted to it. Yeasts grow best under aerobic conditions, but the fermentative types can grow anaerobically, although slowly. Fermentation by yeast is inhibited in presence of air and it is called pasture effect. The yeast cells manufacture two types of hydrolytic enzymes. Extracellular invertase converts sucrose into glucose and fructose, while the intracellular zymase ferments glucose to ethyl alcohol and carbon dioxide. Presence of antibiotic at higher concentration in the culture media have been observed to alter normal phenotype of yeast. For example, production of a true mycelium in presence of penicillin and decrease in the number of mitochondria per cell in successive generations in presence of streptomycin. Now, structure. Sacchromyces are small, unicellular, uninucleate, non-mycelial and non-flagellate fungal organisms. Each cell consists a tiny mass of protoplast enclosed by a definite cell wall. Under electron microscope, the cell wall appears two-layered, an outer electron dense and inner microfibrillar layer, each 0.2 micron thick. According to Mentile et al. 1969 and Webster 1980, the cell wall in Sacchromyces cerevisiae is relatively thick and distinguished into three layers: an outer layer of mannan protein with some chitin, middle glucon layer, and the innermost protein glucon layer. The bulk of the cell wall is composed of beta-glucan and mannan. Glucan is known as yeast cellulose. Chitin in their cell wall is virtually absent. The percentage of different constituents in the cell wall is glucan 30 to 40%, mannan 30%, lipids 8 to 10%, proteins 6 to 8%, and chitin 1 to 2% or even lesser. The plasma lemma tightly adheres to the cell wall and it has increased surface area due to the presence of shallow pit and invaginations. The cytoplasm is granular, containing oil droplets, glycogen bodies, volutin granules, besides the cell organelles typical of an eukaryotic cell, including the ATS type of ribosomes. Transmission electron microscopy has revealed a single giant and branched mitochondrion in a strain of Sacchromyces cerevisiae. However, their number mostly ranges from 50 to 100. The mitochondria possesses 70 S-type of ribosomes in their matrix. The relation of nucleus to the vacuole has remained debatable for quite a long period among the mycologists in the past. But electron microscopic studies of ultra-thin sections of Sacchromyces cerevisiae by Agar and Douglas and Hashimoto et al. Have revealed the nucleus and vacuole as two separate entities. A large vacuole bound by a tonoplast often occupies a considerable portion of the cell. The vacuolar sap contains granules of polymetaphosphate and lipids besides many proteolytic enzymes. A well-defined but smaller nucleus covered by a porous double membrane nuclear envelope lies close to the membrane of the vacuole. A spindle pole body, also called spindle plaque, lies within the nuclear envelope. According to Webster 1980, yeast nucleus can be studied best at the time of budding and it is difficult to be seen in a living cell. The genome of Sacchromyces cerevisiae is very small and the chromosome number is also small, 32 linear chromosomes of 200 to 2200 kilobase. Thus, 2n is equal to 32 and n is equal to 16. The yeasts also show polyploidy. The Sacchromyces cerevisiae genome was sequenced during 1996. The nuclear DNA has a density of 1.699 grams per cubic centimeter and a GC content of 40%. In yeast cells, 10 to 20% of cellular DNA is localized in a single mitochondrion. This DNA has a density of 1.683 grams per cubic centimeter and a GC content of 21%. The role of mitochondria in cytoplasmic inheritance was first understood in Sacchromyces cerevisiae. The mitochondrial mutants of this yeast have smaller cell size and are termed as peptides. They also form smaller colonies as compared to wild-type cells and occur at a frequency of about 1%. The mitochondria of these peptide mutants lack cytochrome oxidase, and hence defective growth. The nuclear division in Sacchromyces is intranuclear. The SPB divides and the two daughter SPBs move to the opposite ends of the nucleus. The nucleus elongates and becomes dumbbell-shaped. The entire nuclear mass is still bound by the nuclear envelope. In late telophase, a constriction in the isthmus separates the two daughter nuclei, and this pattern of division is almost same in mitosis as well as in meiosis. Now comes the reproduction part. Sacchromyces includes true fungi as they reproduce sexually besides the asexual method. Asexual reproduction. It occurs by the process of budding, hence the species of Sacchromyces are known as budding yeast. Budding is the most common method of reproduction and it occurs under favorable growth conditions. Both haploid and diploid cells reproduce by this process. The form of budding in Sacchromyces, where a limited area of the wall becomes plastic and balloons out to form a bud, is referred to as blastic development by Hudson in 1986, and the bud is also called blastospore. The peptide mutants reproduce only by budding and can be maintained indefinitely in the vegetative state, either as haploids or diploids. Buds arise at predetermined spots of the cell wall. Vesicles carrying newly synthesized cell wall material first accumulate just beneath the cell membrane at the budding sites. Following mitosis, the cell wall bulges and stretches out to form the bud. Cytoplasm, together with the daughter nucleus, various other cell organelles and even a small portion of the vacuole flow into this newly formed bud. When the bud is fully formed, a centripetal formation of septum occurs at the isthmus joining the bud and the mother cell. The primary septum made of chitin is first formed, followed by the addition of secondary septum composed of glucan. Separation of the bud from the mother cell takes place and a convex bud scar is evident on the mother cell opposite to a concave birth scar on the daughter cell surface. Daughter cells grow by the overall increase in cell size. A bud scar resembles a crater on the surface of the mother cell with a raised circular rim partially consisting of chitin. Each cell has a birth scar and a number of bud scars. A yeast cell can in time produce 23 to 24 daughter cells by budding, and hence as many bud scars have been seen on a single cell. By counting the number of bud scars on a mother cell, one can know the number of buds that have only been produced from it. Buds arise only from the unscarred regions of the cell wall. When a mother cell has no more unscarred cell wall remaining, it can no longer reproduce and will senesce. The generation time of Sacchromyces is very brief, only about 90 minutes. If the nutrient supply is available in abundance, the budding process is very quick, producing a branched or unbranched chain of 60 to 100 cells called the pseudomycelium or pseudohypha. The cells in a pseudomycelium are loosely joined together and independent, not interconnected by pores. Soon the chains break and the constituent cells are separated from each other. Now, sexual reproduction. Sexual reproduction in Sacchromyces is extremely simplified with three phases: plasmogamy, karyogamy and meiosis. Sexual fusion called hologamy takes place between two ordinary haploid somatic cells of opposite mating types. The vegetative cells function as gametangia and their protoplasts as gametes. Sacchromyces show bipolar and unifactorial heterothallism. The two mating types are especially controlled by a single gene which exists in two allelic states, A and alpha. Legitimate copulation called conjugation or gametangial conjugation occurs between two isogametangia. The isogametangia produces sex hormones or pheromones to attract the opposite mating type. These are referred to as A and alpha factor. The alpha factor has been purified and found to bear a molecular weight of 1400. The two isogametangia pair up and develop a conjugation tube. Plasmogamy results in the formation of a temporary dikaryotic protoplast, wherein the karyogamy immediately results in the formation of a diploid zygote. If the conditions are favorable, the zygote behaves as a large strain somatic cell and reproduces by budding. But under the shortage of water, food and low temperature, the large strain cell or even the zygote functions as the thin-walled ascus. Sacchromyces cerevisiae is a holocarpic fungus. As the unicell develops into a sporangium, the ascus. Ascocarp does not develop, that is, the ascus is naked. Meiosis in the ascus results into 4 haploid ascospores, 2 belonging to each mating type. These acid-fast spores are globose or ovoid and are only slightly more heat resistant than the vegetative cells and will normally be killed by temperature at or below 100 degrees Celsius, example in the baking of bread. The ascospore wall is differentiated into 3 layers. The middle thick electron transparent layer enclosed in between 2 thinner electron dense layers. The mature ascus wall deliquesces, releasing the ascospores. They germinate only under the favorable growth conditions to form the progeny of haploid yeast cells that grow in number by budding. The petile diploid cells cannot sporulate, nor can a mating between 2 haploid petites produce a zygote that will sporulate, and hence they also do not show the alternation of generations. And now the alternation of generation. In Sacchromyces cerevisiae, the large strain yeast cells represent the diplophase or sporophytic phase. In addition to vegetative reproduction by budding, they, under certain conditions, directly function as asci, and each ascus produces 4 ascospores meiotically. These ascospores normally germinate to produce the haploid dwarf strain of the yeast, which represent haplophase or gametophytic phase. The gametophytic cells bear no sex organs, and under certain conditions they themselves function as gametangia. Their protoplasts function as gametes. The gametangia resort to conjugation and fuse to form the diploid zygote. Zygote by budding give rise to large strain diploid yeast cells representing the diplophase. It is evident from this account that the life cycle of Sacchromyces cerevisiae consists of 2 distinct phases, the sporophytic phase or diplophase and the gametophytic phase or haplophase. The former consists of the large strain diploid cells and the asci, while the latter consists of the ascospores and the dwarf strain yeast cells, and these 2 phases alternate in a single life cycle. It is interesting to note that in the life cycle of Sacchromyces cerevisiae, both the haplophase and the diplophase are of equal importance, and each phase is perpetuated by budding. Such a type of life cycle or alternation of generations is known as haplodiplobiontic or diplohaplobiontic life cycle. With this, we conclude our today's program that was about important features and life history of Sacchromyces. Thank you.